This is the second video lecture for microbiology for October 10th, 2019. And where we left off, we were talking about aerobic respiration, and we should also mention the different types of anaerobic respiration. The first one is denitrification, and with denitrification, nitrate is the terminal electron acceptor. Reaction looks something like this, where nitrate is converted to nitrite and water. Also, you can have sulfate as the electron acceptor in sulfate reduction. And in this case, sulfate is converted to sulfite and water. And then finally, carbon dioxide or carbonate ion as the electron acceptor in methanogenesis. Methanogenesis happens by an entirely different pathway that you don't really need to worry about in the scope of this class. Also, in the absence of a terminal electron acceptor, on when that happens, okay, glucose will ferment directly, and it will incompletely oxidize into either ethyl alcohol or lactic acid. Lactic acid will occur in homolactic bacteria as well as in human muscle. Alcohol will occur in some bacteria and also yeast. And we define fermentation as the incomplete oxidation of glucose in the absence of oxygen. Like the previous slide stated, it produces only 2 molecules of ATP from glucose. And there are 2 types of fermentation. One is an alcoholic fermentation with yeast and some forms of bacteria, converting pyruvate to ethanol. And the other is acidic fermentation, which is primarily lactic acid bacteria, converting pyruvate to lactic acid. There are also other types of bacteria that will produce a mixed type of acid. This is called mixed acidic fermentation, and this will produce a common combination of acetic acid, lactic acid, succinic acid, and formic acid in order to lower pH. Here's a fermentation of yeast, and it is occurring in grape juice to make wine. This is a type of yeast called Saccharomyces cerevisiae. It's a friendly yeast. You can't really get a yeast infection from Saccharomyces cerevisiae. And here is what happens when you exceed your oxygen capacity and while you're working out, and your muscles start to burn. The reason why your muscles are burning is because you're producing lactic acid. There is no terminal electron acceptor, there's no available oxygen, and so the glucose is directly fermented into lactic acid. It also occurs in lactic acid bacteria. This is a lactobacillus that produces lactic acid. Now, when we look at catabolism in general, and you look at aerobic respiration, the impression is that glucose is the only thing that can be broken down for energy. And that's not true. We know that from a dietary perspective that fats can be broken down as well as proteins, and any of those items can be broken down in order to gain energy. Fats are broken down into dihydroxyacetone phosphate, and hydroxyacetone phosphate can enter into glycolysis. Proteins are broken down to amino acids, and then they're deaminated. The amino group is taken off to produce carbon compounds that go directly into the Krebs cycle. Okay, so if we look at that perfect food here, which is pizza, pizza has protein, carbohydrates, fats, and oils. And these different groups can enter in different places into either glycolysis preparatory reaction, the Krebs cycle, or the electron transport chain. And in these instances, then they can be utilized to produce energy via ATP. So proteins, like I said before, are converted to amino acids. They're deaminated, so you get rid of the amino group, and they can enter into glycolysis as pyruvate. They can enter as acetyl-CoA, or they can enter directly into the citric acid or the Krebs cycle. Fats can be converted to glycerol or fatty acids. Glycerol then can be metabolized into dihydroxyacetone, which goes into glycolysis. Fatty acids can be converted directly to acetyl-CoA. So let's review. What's ATP's role in the cell? Well, ATP is the energy currency of the cell. This is how energy is distributed throughout the cell. And this is once energy is depleted, that converts ATP to ADP, and then ADP is regenerated to ATP through the electron transport chain. NADH's role in the cell is just an electron carrier. It carries electrons to the electron transport chain so they can be converted into energy-rich ATP. The three components of aerobic respiration, and really we should say 4 components here. So I want you to scratch that out and put 4 components of aerobic respiration. The first one is glycolysis. The second one is the preparatory reaction. The third one is the Krebs cycle. And the fourth one is the electron transport chain. In fermentation, we have defined as the incomplete oxidation of glucose in the absence of an electron acceptor. Denitrifiers, they breathe nitrate, they reduce nitrate to nitrite in order to get energy. Okay, and then here's an interesting concept. This is amphibolism. Amphibolism is when both anabolism and catabolism are occurring at the same time. So amphibolism is both processes that occur in order to improve cell efficiency. And some important processes that can go both ways. Instead of glycolysis, you can have gluconeogenesis, which is the formation of glucose. Instead of the buildup of lipids, you can have beta oxidation, which is fat degradation to acetyl-CoA. Instead of deamination, you can get amination to form amino acids from compounds like pyruvate. And then you can also have transamination, where you transfer amino groups to form new and different amino acids in order to have the complete version of 20 amino acids for the production of proteins. So glucose can go in a variety of directions. You see the central metabolite pyruvate here, pyruvic acid. And all of these different products can go either towards energy generation through catabolism, or up through the top, you can have amino acids converted to nucleotides for nucleic acids, and amino acids from pyruvic acid and acetyl-CoA form proteins. You can have carbohydrates that can form starch and cellulose for cell walls. And then you can have fatty acids for lipids and fats and for membranes. In terms of carbohydrate biosynthesis, these are anabolic processes. You can have glucose 6-phosphate form a polysaccharide called glycogen. Glycogen is stored in the muscles. You can have peptidoglycan, which is a cell wall, the component of the cell wall in bacteria. It's formed from glucosamine and muramic acid. Nucleic acids can be formed from carbohydrates, and glycocalyx is formed via polysaccharide biosynthesis. Proteins are synthesized from 20 different amino acids, and DNA and RNA biosynthesis we'll cover in Chapter 9. Now, the other major process that we focus in on in Chapter 8 is photosynthesis. Photosynthesis is the generation of glucose as an energy source from sunlight. Okay, so what is required for photosynthesis is the formation of a carbon energy source like glucose, and it has to be converted from sunlight. A byproduct of photosynthesis is the uptake of carbon dioxide to make oxygen. Not all types of photosynthesis make oxygen, but all photosynthesis does take light energy in order to make a carbon source like glucose. And with very few exceptions in our world, in our biosphere, sun provides energy to drive life processes. So there are light-dependent reactions that require the presence of sunlight, and these are primarily catabolic. And there are light-independent reactions that proceed regardless of the light conditions, and these are primarily anabolic. Light is propagated as photons, and these are little packets, they're massless packets of light. They're energy, and they travel as waves. And as light hits photosynthetic pigments like chlorophyll, some of the wavelengths are absorbed as energy and others are reflected. What we see reflected is the colors of things like plants, which are green. So the pigments that can carry out some types of photosynthesis are chlorophyll, which is green, carotenoids, which are yellow, orange, and red, and phycobilins, which are red or blue-green. And so when we look at the process of photosynthesis, here's a chloroplast. So obviously this is a eukaryotic cell if it has a chloroplast in the first place. So instead of having oxygen and glucose entering in, we have water. Water is split to form oxygen, and then we have carbon dioxide, which comes in and combines with hydrogen and electrons, and that forms glucose coming out. So really, this is the reverse process chemically from respiration. And photosynthesis can be separated into light-dependent reactions and light-independent reactions. First, we'll discuss light-dependent reactions, which occur in the grana of the thylakoids, in chloroplasts for eukaryotic cells, or in the cell membrane for prokaryotic cells. Electrons are generated by splitting water, this is called photolysis, into protons and oxygen by a protein complex called photosystem II. So the first thing that happens in photosynthesis is water is split via the action of photosystem II. Photosystem II excites electrons to form ATP, and then the electrons are carried through photosystem I, where they are excited a second time, and they are used to reduce NADP or NADP to the electron carrier NADPH. There is a proton gradient inside the thylakoids. This is caused by photosystem II, and this generates ATP via ATP synthase. So it's like there's a little electron transport chain that occurs in photosynthesis, and because of that proton gradient, then we get ATP. That ATP is not used in the cell. It's only used for the production of glucose during photosynthesis. Okay, so if you follow this around, You have photosystem 2, the electron transport chain, and photosystem 1 are light-dependent reactions. These occur in the thylakoids, either within the chloroplast for eukaryotes or in the plasma membrane for prokaryotes. Photosystem 2 takes water and it converts it to oxygen, and then the excess electrons and hydrogens are carried by NADPH. Photosystem 2 creates ATP. Photosystem 1 excites electrons to create NADPH. Then this goes to light-independent reactions, which occur during sunlight and darkness, called the Calvin cycle. In the Calvin cycle, carbon dioxide is taken up and along with additional water. This makes G3P, which is glyceraldehyde 3-phosphate, which is converted further to glucose. Through this process, energy is used, so ADP is formed, and electrons and hydrogen are dispersed, so NADP plus is formed. Okay. So light-independent reactions are the Calvin cycle, and this is where carbon dioxide is fixed within the chloroplast or within the thylakoids. And carbon dioxide is combined with a 5-carbon acceptor. This is called carbon fixation. This results in a 6-carbon intermediate, and it splits into 2 3-carbon molecules called 3-phosphoglyceric acid. The resulting reaction in the pathway generates fructose, which is then converted to glucose. And if you see this reaction, what happens is carbon dioxide then combines with RUBP. RUBP is ribulose bisphosphate. It has 2 phosphate groups in it. Combines with carbon dioxide. This makes 3-phosphoglycerate. Uses ATP, and it also gets electrons and hydrogen from NADPH. And then this produces phosphoglycerate is reduced to glyceraldehyde 3-phosphate, G3P. G3P combines with another G3P, and that forms glucose. Glyceraldehyde 3-phosphate then regenerates ribulose bisphosphate, and the cycle continues. Now, I will be very clear in terms of what is required on the exam. The enzyme that is used in order to fix carbon dioxide in photosynthesis is rubisco, which is ribulose bisphosphate carboxylase, but we just call it rubisco. It's very important, and it's the most highly abundant enzyme that you find in plants and also in chloroplasts and thylakoids and microbiology. But I really want you to know these 3 steps. You don't need to know every name, but you do need to know that carbon dioxide is reduced via the action of rubisco. You do need to know that 3-phosphoglycerate is reduced to glyceraldehyde 3-phosphate. And then you also need to know that ribulose bisphosphate is regenerated from glyceraldehyde 3-phosphate. Okay, so you need to know those 3 steps. You also need to know what rubisco is. Okay, so first of all, what is amphibolism? Amphibolism is the fact that catabolic and anabolic processes go on at the same time. Some are direct reverse of each other, like gluconeogenesis and glycolysis. They go on at the same time in order to optimize cell metabolism. The main cellular anabolic processes all correspond with the macromolecules. So the macromolecules would be lipids, proteins, amino acids, and nucleic acids, and the main cellular anabolic processes are just the processes by which you get lipids, nucleic acids, amino acids, or proteins, and carbohydrates. Which generates electrons, photosystem 1 or photosystem 2? Photosystem 1 generates electrons, and photosystem 2 generates energy. Okay, so photosystem 1 is where we get NADPH, and photosystem 2 is where we get ATP. What does the Calvin cycle do? The Calvin cycle fixes carbon dioxide in order to form glucose. It also uses ATP and NADPH. In the first step, carbon dioxide is fixed. In the second step, 3-phosphoglycerate is converted to glyceraldehyde 3-phosphate. And in the third step, glyceraldehyde 3-phosphate is regenerated into ribulose bisphosphate. Those are the 3 steps. Okay, and that concludes the lecture for Chapter 8. Just going to check on the time here, and we will pause. Chapter 9 is a chapter regarding microbial genetics, and our objectives in this chapter are to look at the basics of DNA structure and how DNA replicates, and then to understand 2 other processes. The first one's transcription, and the second one is translation, and these processes lead to the creation of protein. So there are 3 separate processes here, and students often conflate these processes. They'll take portions of replication and they'll apply it to transcription and translation. And what I want you to do is just separate out these 3 processes in your mind. Replication is where DNA duplicates to form new cells. Transcription is where DNA is transcribed and read to make messenger RNA. And translation is where messenger RNA is read and translated into protein. Then we'll talk about different genetic recombination phenomena. Remember, when we're talking about prokaryotic cells and other cells that reproduce by binary fission, their only method to acquire genetic diversity is through mutation. So we have to understand how these genetic materials recombine in order to create mutation, in order to create genetic diversity for the species to survive. Okay, so biological traits are held in the genetics. They're transferred from parents to offspring. Genetics is the expression and variation of these traits. Genetics is also the structure and function of genetic material and how this material changes. When we talk about the genetics of the entire organism, we'll talk about organismal genetics, and that's the heredity of the whole organism or cell. When we talk about the genetics of individual chromosomes, we call that chromosomal genetics. And molecular genetics is the biochemistry of the genes themselves, how the genes replicate, how the genes transcribe and translate. So if we have eukaryotic cells over here, then eukaryotic cells have chromosomes which are linear, and their linear chromosomes are encased within the nucleus. They also have chromosomes within their mitochondrion and their chloroplasts. Okay, and some will have plasmids. In some fungi and protozoa, you'll find plasmids as well. In prokaryotic cells, there is no nucleus, so the chromosome is exposed. It's circular, and it's in a region called a nucleoid. Note that eukaryotic chromosomes are linear, prokaryotic chromosomes are circular, and prokaryotes do have plasmids. So that includes bacteria and archaea, which have plasmids and circular chromosomes. Generally, prokaryotic cells have a single circular chromosome. And then we have viruses, which can be DNA double-stranded or single-stranded, and then RNA double-stranded or single-stranded. If we look at the sum of the genetics of an individual organism, we call that sum the genome. This includes chromosomes as well as non-chromosomal sites, what we would consider plasmids, and the DNA in the chloroplasts and the mitochondria. Genomes of cells, cellular material, has DNA, but in viruses, they may include either DNA or RNA, since viruses can be DNA-based or RNA-based. A chromosome is just a discrete cellular structure composed of neatly packaged DNA molecules. So chromosomes are not organelles. They are molecules. You find chromosomes in both prokaryotic and eukaryotic cells. Eukaryotic chromosomes are linear, and the DNA molecule is tightly wound around histone proteins located in the nucleus, as we showed before. Some cells can have a few chromosomes, whereas some species, like orchids and lilies, may have up to 100 chromosomes per cell. Eukaryotic chromosomes can occur in pairs or singles. The pairs we call diploid, and the singles we call haploid. Prokaryotic chromosomes are circular. They're secured via histone-like proteins. They are in a discrete packet called the nucleoid, but there is no nuclear membrane. And most bacteria just have one, so we find a single circular chromosome. A gene is just the fundamental unit of heredity responsible for a given trait of an organism, and this is defined in classical genetics. And in molecular genetics, we define it as a site on the chromosome which provides information for a cellular function. And the gene carries information to synthesize a protein and or an RNA molecule. Typically, a gene is transcribed to RNA and then translated to protein. There are 3 different types of genes. There are structural genes that always code for proteins. There are genes that code for RNA only, and they do not lead to proteins. And there are regulatory genes that control gene expression. An individual's genetic makeup is their genotype as opposed to their phenotype, which is their outward appearance. And phenotype is related to genotype, and it may change based on which genes are turned on. The smallest genome in cellular life is E. coli, with only 4,288 genes. Human cells have anywhere between 20,000 to 25,000 genes. That's still being discovered as a part of the Human Genome Project. Now, if we look at DNA, isn't that cool? My DNA molecule rotates. But if you look at DNA, it is a molecule called deoxyribonucleic acid. Okay, and the backbone, there are 4 different molecules of DNA. One's adenine. What binds adenine is thymine. Another one is guanine, and what binds guanine is cytosine. There are 2 distinct types of bonds. There's a phosphodeoxyribose backbone, which are phosphate bonds on either ends. These are the rails of the ladder. And then there are hydrogen bonds between the bases on the rungs of the ladder. And you can see the hydrogen bonds. There are 4 types of DNA nucleotides: adenine, thymine, guanine, and cytosine. And each nucleotide is made of a nitrogenous base, ribose sugar, and a phosphate group. This ribose sugar is called deoxyribose because it's missing an oxygen from this carbon here. So if we have phosphate, deoxyribose, and nitrogenous bases. A double ring base is called a purine, and a single ring base is called a pyrimidine. Purines always bind pyrimidines, so A always binds T, and C always binds G. So when we have our phosphate bond ring, or rung, our phosphate bond rail of the ladder, we have adenine, it binds thymine, cytosine binds guanine, and so forth. And each rail of the ladder is going in an opposite direction. We always read from 5' to 3'. So this rail of the ladder is going down, and this rail of the ladder is going up. When this arrangement where one rail of the ladder is read up and the other one is read down is called the anti-parallel arrangement. And like I said, the direction of DNA is always 5' to 3'. So we start at the 5' end, and then we work our way to the 3' end. And students always wonder, what does this 5' mean? This is just the phosphate that is attached to the end, the 5' carbon, and then we just count the carbons on deoxyribose, 5, 4, 3, 2, 1. Those are the carbons on deoxyribose, and we'll start reading at the 5, and we'll go to the 3. Then we'll hit a phosphate, then we'll read the 5 and go to the 3, and hit a phosphate, and so forth. Down here, we go from 5 to 3, and then phosphate, 5 to 3, then phosphate, and so forth. So that's where the 5' and 3' come. Those are just carbons that are on the deoxyribose itself. DNA is significant in that it maintains its code during replication or reproduction. So the code will be retained during cell growth and cell division. And when the two strands are separated, each stands as a template for the other one. DNA is also very abundant. The length of DNA gives rise to numerous combinations, and the DNA code maintains unique qualities for each organism. When DNA replicates, we call it semi-conservative replication. And the way that works is you've got a DNA molecule that unwinds and unzips through the action of an enzyme called helicase. That exposed different strands, what we call the parent strands in blue of DNA. Then an RNA primer is put in place, and you get DNA polymerase molecule here, which is this sort of molecule that looks like an oval, and DNA polymerase here that looks like an oval. This is going from 5' to 3'. This is also going from 5' to 3'. And then as DNA polymerase progresses down, then that creates a new strand, which we call the daughter strand. The daughter strand is on either side and is always accompanied by the original strand of DNA in blue, which we call the parent strand. So we call this semi-conservative replication because each daughter molecule, which is shown in pink, will be identical to the parent, which is shown in blue in composition, but neither one of these strands here on this side or on this side are completely new. They're always part parent and part daughter. So replication starts at the ori, which is the origin of replication. And this is a short sequence of DNA that you find on the chromosome that's always rich in adenine and thymine. So it's AT rich. And AT is easier to get apart. It's easier to separate those rails of the ladder apart because they're only held together by 2 hydrogen bonds rather than 3 for guanine and cytosine, and therefore it's easier to separate. At the origin of replication, helicase will untwist the helix and breaks the hydrogen bonds, and so it exposes single strands of DNA. Then DNA polymerase III synthesizes new daughter strands of DNA using the parental strand as a template. Okay. Now, for DNA polymerase III to work, the DNA must be unwound and separated by helicase. DNA polymerase is unable to initiate the replication process without an RNA primer. Okay, so the RNA primer binds at the origin of replication, and then DNA polymerase can add nucleotides only in one direction, from 5' to 3'. Now, in circular chromosomes, the way this happens is you have the origin of replication at the top of the DNA strand. The two strands of DNA then unwind and unzip from helicase. DNA polymerase then orients to go in the opposite directions. One is going 5' to 3' on the top strand. The other one's going 5' to 3' on the bottom strand. Where these strands open up, we call those replication forks. And then DNA polymerase progresses 5' to 3' on this strand, 5' to 3' on this strand. And the leading strands are duplicated just nucleotide by nucleotide. And then the lagging strands, which are opened up at this point here. This is the lagging strand because it's 3' to 5'. This is the lagging strand here, it's 3' to 5'. These duplicate via fragments of DNA called Okazaki fragments. So the lagging strand does not use DNA polymerase III, it uses these Okazaki fragments that come together here. These are just small fragments of DNA, just small nucleotides, and these are put together via an enzyme called DNA ligase. DNA ligase is a DNA glue, and it just glues the different nucleotides together. So on the leading strand, you have DNA polymerase III that goes from 5' to 3' and replaces a single nucleotide at a time. On the lagging strands, the DNA is populated with Okazaki fragments, and the Okazaki fragments are put together using DNA ligase. So the RNA primer is synthesized and enters at the ORI. DNA polymerase III uses this primer as a starting point to add DNA nucleotides. The helix structure opens up via helicase, and that forms replication forks. The leading strand is synthesized continuously, just a single nucleotide at a time, using DNA polymerase III. The lagging strand is populated simultaneously with Okazaki fragments, which are small fragments of DNA, and these are glued together via DNA ligase. DNA polymerase I removes the RNA primers and replaces them with DNA. And when the replication forks come for full circle, then the DNA ligases link the fragments together and they form two separate daughter molecules. Now this is for circular DNA, which you would find in prokaryotic cells. Okay, so this whole concept of these circular DNA components acting together for leading strands and lagging strands is reserved for prokaryotic cells. DNA polymerase III and I also proofread and replace incorrect unmatching bases. If there was not proofreading, there would be an error every 1,000 nucleotides, and so those errors do have to be proofread out. Okay, so what's the difference between organismal genetics and molecular genetics? Organismal genetics deals with the entire genome, all of the traits of the entire organism, whereas molecular genetics are based on a single gene, and genetic structure deal with the way that gene is structured, is duplicated, is transcribed, and translated. Which eukaryotes have plasmids? This would include fungi and protists. What's the difference between a genotype and a phenotype? We've answered this before. The genotype is your genetic structure, and your phenotype is your outward appearance. What does the enzyme helicase do? Helicase unwinds and unzips DNA. And you should be able to describe DNA replication. I've talked about DNA replication for linear DNA as well as for circular DNA, and so you should be able to describe that in detail. Make sure that you can, and there are key slides where each step is broken down.